Moving into section 18.2, we're going to be looking at gene expression a little bit further. We're going to be starting by looking at questions 18 and 19. Question 18 in your book is asking about differential gene expression. What does that mean? Well, what we need to know about that first off is that every single cell in an organism contains the exact same DNA. There are no differences. So in one cell, if there's a gene for whatever, there's the exact same gene in every other cell of that organism. Now, why then are, like, say, heart cells and not kidney cells when they have the exact same DNA? Well, that's a result of differential gene expression. Essentially, what's occurring in differential gene expression is genes on the chromosomes are expressed in some cells, but those genes aren't expressed in every single cell. In other words, a cell can kind of dictate which genes in the genome are expressed and which are not expressed. In multicellular organisms, this gene expression is essential for cell specialization. In other words, it allows that heart cell to be a heart cell and function entirely different than that kidney cell. The cells of our body, however, only express 20% of the genes that are found within that cell, which is your answer to question 19. It's 20% of all those genes. So what controls which genes are expressed and which are not expressed? Well, the common point of gene expression in eukaryotic organisms is in transcription, particularly items known as transcription factors. Transcription factors, their job is to seek out specific sections of DNA genes which are to be expressed. So the transcription factors, they find sections that are supposed to be expressed, and then they make sure that those genes are expressed in a particular cell. I guess that's actually your answer for question number 20, the common point of gene expression. That would be those transcription factors. So transcription factors would be 20. So what exactly controls outside the cell what genes are expressed within it? Well, looking at this image, we can see an initial signal up at the top here that's outside of this cell. Looks like this would be the nucleus right here, this dark blue region, and this would be the rest of the cell, the cytoplasm, this tan region. So this is an external signal that's entering the cell. And the initial signal that we see at the top could be a variety of things, depending on what needs to be produced from the DNA. What proteins do we need to make? Sometimes the signal could be in the form of a hormone. Other times it could be in the form of an inducer, as we saw with operons. The gene that is selected for transcription is located by those transcription factors that we mentioned on the previous slide. So it finds a section of DNA, the transcription factors do, and they go through the process of transcribing it, cutting out the introns, leaving the exons, putting on the cap and tail, and allowing it to leave the nucleus for translation, where it will make that polypeptide and then will be changed into an active protein. Remember, we talked about some modifications that need to be done to proteins to allow them to fold up into a functional state. Moving into question number 21, it talks about chromatin structure, and it's looking specifically in the question for the difference between heterochromatin and euchromatin and their structure and activity. We'll recall heterochromatin is tightly packed chromatin, and because it's tightly packed, it can't be read. So picture a textbook, but the words are like written in 0.25 font, very, very small. You can't read it. But if we enlarge it to, say, 16 or 20 font, now the text is very, very large, and it's capable of being read. In fact, the text on the screen here is size 30 font, so we can read it quite easily. But if, like I said, if it was at 0.25, we couldn't read it. It'd be too small. So heterochromatin would be that 0.25 font. It's very, very tightly packed, very, very condensed, and it can't be transcribed as a result because it's too tightly packed. Euchromatin, on the other hand, is where the genes are looser and spread out a bit more, and because they're spread out a bit more, they are able to be transcribed. So euchromatin kind of refers to true chromatin because it is something that can be transcribed. Now, moving on from that, what changes chromatin from the hetero to the euchromatin states? Well, these are chemical modifications to the histones, which are the proteins that the DNA kind of wraps around as it's coiling up. So let's take a look at some of these modifications in a little bit more detail, and this will lead into questions 22 and 23. So a little bit of stuff that we've talked about previously with histones and DNA. The DNA coils around the histones, which allows them to shorten into nucleosomes, which we had described in class as beads on a string. These regions are unavailable for transcription, the DNA that's wrapped around the histones. And that's because right around those histones it's wrapped kind of tightly. But the DNA that's found between the histones, that can be transcribed. Nonetheless, the DNA wrapped around the histones, it still needs to be made available for transcription. So they do need to be made available. Histone acetylation refers to the loosening of those regions of DNA so that they can be transcribed. So histone acetylation, that will loosen the DNA that is immediately wrapped around those histones, allowing that DNA to be transcribed. I'm going to explain this in further detail on the image on the following slide here. So here's that image. Histone acetylation refers specifically to the addition of acetyl groups to the histone tails, and we can see those tails right here. So we need to add some acetyl groups to those tails. These acetyl groups attach to lysines, which are amino acids found on the tails. So there's lysines that exist here, and we need to add acetyl groups to them. This image doesn't show us those lysines, however, which is kind of unfortunate, but we can see on the bottom image that the nucleosome on the left, nucleosome right here on the left, becomes uncoiled, so it becomes uncoiled or loosened with the addition of the acetyl groups. Once the histones have been acetylated, the chromatin can further expand and contract with the attachment of either methyl or phosphate groups to the histone tails. So we can acetylate them and we can cause them to spread out a bit, and we can add, from this point right here, we can add methyl groups or phosphate groups, which cause them to either A, loosen even more, or B, tighten up from this state. So acetyl groups, once again, loosen this into this. Methyl groups and phosphate groups either loosen them more or contract them more from this particular state. Specifically, those phosphate groups are the ones that will cause the DNA to loosen even more. Adding methyl groups to the histone tails will cause this to tighten up a bit, so it will become less readable in terms of transcription. In the case of adding methyl groups, causing this to tighten up, that is called DNA methylation, which is question number 23. It's where we add methyl groups to these histone tails. You can kind of see those little green dots there. Those green dots would represent the acetyl groups. So if we add methyl groups to that and methylate it, that's DNA methylation. These histones, they tighten up again. It appears that the attachment of items to the histone tails, once again, our histones are these purple things, the tails are right here. It appears that attachment of these items to the histone tails plays a significant role in gene expression. There's actually a term for this called the histone code hypothesis. The histone code hypothesis proposes that specific combinations of modifications to the histone tails ultimately determines the shape of chromatin. Thus, it controls what can and cannot be transcribed, which is also part of question number 23. We talked about adding a lot of things to these tails, and that's the histone code hypothesis. Which combination is added to these tails? Well, that determines if this strand can or cannot be transcribed. Moving into a little bit more detail on that DNA methylation, or adding methyl groups to the histone tails, questions 24 and 25. DNA methylation is the addition of those methyl groups to certain bases in DNA is associated with reduced transcription in some species. And that's because the DNA is more tightly packed with those methyl groups. And when the DNA is tightly packed, it cannot be transcribed. We see this in the human X chromosomes. In females, well, keep in mind males, females. Females have 2 copies of the X chromosome. Males only have 1 copy because male sex chromosomes are XY. In females, only 1 of those 2 X chromosomes in every cell is expressed. The other cell is not, or the other X chromosome is not expressed at all. In other words, the second X chromosome is kind of shut off. This is called X chromosome inactivation. It occurs when the inactive chromosome is heavily methylated, and as a result, it cannot be transcribed. And that's what number 24 is looking at. So that X chromosome inactivation, as a result of DNA methylation, would be an example of a long-term inactivation of a gene. Pretty much that X chromosome that's turned off, it will never be turned on for the life of that female. Genomic imprinting, on the other hand, number 25, is a methylation that regulates the expression of either the maternal or paternal alleles in certain genes at the start of development. So we're looking at genomic imprinting. This is occurring right at fertilization, those very, very early developmental stages of an embryo. And this will determine which alleles are expressed in individuals, which is why you'll have some children that look a little bit more like their mother versus their father. That's because those genes from the father might be turned off, meaning that the ones from the mom are a little bit more turned on. I've had some friends before, one family in specific I was friends with when I was growing up. Mom and the older daughter and the younger daughter who I was friends with, all 3 of them were like twins. It was remarkable, but there was a separation of years between each of them. So that's one extreme case where the maternal genes are definitely more present than the paternal genes. And in the beginning of our next discussion, I'm going to take you to this website here, which is, it's cool. I really like this site. It's talking about epigenetic genes and epigenetic inheritance, which I'm going to describe slightly differently than the book did. Epigenetic genes are things that are sections of DNA that you can turn on or not turn on. And in some cases, there's critical moments in development where if a gene is not turned on, it's going to be permanently turned off. So like you can almost start to think like psychopathic killers here. Possibly could be psychopaths as a result of epigenetic inheritance or epigenetic genes, which were not turned on at very, very critical points in inheritance. Now, I can't really say if that's true or not for serial killers, but when you see this sample that I'm going to take you through in the next discussion, you might understand why I would maybe think that. So we'll call it here, and we'll pick up on our next discussion.